Donc, j'ai pas Jacques.
English.
Enfin, Jacques.
The title was
English.
in English, so I will give
Okay.
the presentation in English.
Okay, so I will introduce you in English. So Jacques is a professor at the University of Aix-Marseille. He has been involved for a long time in projects for development of algorithm and genomic resources, for example, the famous ERSAT server. And since 2017, I think, he's a co-director of the French Institute of Bioinformatics, and Jacques and Claudine present you on Tuesday this institute. And he will present you today the EMERGEN project, which was initiated a few days ago, in fact, which aimed to setting up a platform for genomic monitoring and COVID-19 research. So, Jacques, it's up to you.
Yep. Can you see the slides?
Yep.
Yep. Okay, so thank you for the introduction. Actually, the project started not a few days ago, but a few months ago. It was end of January, but nevertheless, it has been a very fast project. So I put here all the people who contributed directly to the project. There are many more people, but those who contributed to the bioinformatics platform. So the project actually was launched somehow urgently by the French Ministries of Health and the French Ministry of Research, and they asked the Santé Publique France and the ANRSMIA to launch ambitious genomic project from one day to the next one, and it had to start very fast. And so there is a consortium that is being formed, including Santé Publique France, four big platforms of sequencing associated to the National Center of Reference for Viruses, and then a network of 43 platforms who were initially created for the HIV monitoring and who now extended their scope to all the infectious diseases. And there was an urgent need to have one partner that was able to set up a platform of bioinformatics that would host viral sequences, plus also the health-related data. And so this is what I will present. And it is a collaboration between IFB and the INSERM and the National Center of Storage and Computation, the CINES in Montpellier. So the goals of this EMERGEN was to have a sequencing capability of 10,000 genomes a week in order to follow the recommendation of the WHO, and to be able to identify variants of concerns, variants of interest, but also those that were already described in the international repositories, but also to detect novel variants, to monitor the variant evolution in France, and also to do the pairing between the genome sequences of the viruses and health data. And this is a difficult challenge, because this means we have to work with a highly secure environment. And also there is the use of the result has several purposes. So here you see a sort of a schema of the data flow. So the network regroups more or less 70 labs who are doing the sample collection. And then these labs send the samples to the sequencing platforms. And there are about 50 sequencing platforms, because this NRS regroups 40 platforms. In addition, now there are some private labs who are joining. And all the sequences have to be deposited in this bioinformatics platform that we are developing now. And then once we do the analysis, we host both the viral sequences, which are non-sensitive, and some health data, which is sensitive. And we organize the results in a database called EMERGEN-DB. And from this database, we can provide the results to surveillance via Santé Publique France, but also it must be accessible for research and all the sequences as well. And there is a work package in the project with 14 different research projects related to making the best use of this data, and for instance, to detect new variants, to predict the impact of variants, to do some structural analysis, to detect variants in used water, and so on. And all this has to be used also for care and prevention. An important aspect of the project also is that all the data that is produced has to be open science. And so we will submit it both to GIZ, which is not open science. It's this data that is shared between the community of virologists with a protection, and ENA, which is the European the European Nucleotide Archive, with whom we are collaborating as the French node of Elixir. So we developed the project in 3 phases. The first phase is the handling of non-sensitive data about variant, and basically it is about it's not the sequence, it is just the annotation of the variant. And this has been developed in a few, really in a few days, literally, with the strong involvement of the NNCR, of all the colleagues of IFB who participated in this, and of Thomas Denecker, who developed a database in Django. So this is the architecture of this database that was developed by Thomas, and also that was deployed with the help of the mutualized task force of the NNCR. And this is based on Django as the main database management system, and the storage, the backend is with PostgreSQL. Then there is a lot of different components. I have no idea what they do, and I have no time to develop it nevertheless. Then based on this backend, there are inputs and outputs, and the idea was to make it easily accessible for biologists. So what Thomas did is to develop a front end that enables to upload Excel files. We defined together with Santé Publique France really strict formats for the input. And then once the data is there, there is a conformity control performed by the database, and there is a mail sent to the submitters. And this mail, if there are some mistakes, inconformities, invalid dates, or so on, the mail just gives the list of all the errors. So there is an annotation of each row of the input telling this is correct, this is incorrect, this is incorrect, and the reason why it is incorrect. And then the people correct the mistakes and they resubmit the data until it is completely conform. And then this is stored in the database. Once it has been stored in a database, you can export it in different formats. And first, you can download Excel files with the front end, but you can also have an API that enables to export the data in JSON and to do more specific queries. Then there are different ways of visualizing the data and so on. And for instance, now I show you here some of the stats page that shows the number of entries currently stored in the database, the submissions per month, and for each of the groups of sequencing platforms with the National Center of References, the ENRS network, and other platforms are starting now to submit since last week. And these are private laboratories. And you can also have some visualization of all the distribution of the variant in France. The second phase is starting since a few weeks. We started preparing it, and it's starting right now. And this is the upload of the sequences, then the analysis, and the deposition in international repositories. So for this, there is already a Galaxy server that has been set up by the Galaxy team of France, of IFB. And they also took advantage of all the workflows that have been developed by the international Galaxy community for the SARS-CoV-2, so SARS-CoV-19. So all this has been, SARS-CoV-2, sorry. So COVID-19. So this has been installed on the server. It's available right now. And then above this Galaxy server, there is a need to handle all the data fluxes, because the platforms has to deposit tens of thousands of sequences. So they cannot do this with the graphical interface of Galaxy. And so what has been now developed is what we call a meta workflow that is based on the organization of the orchestration of all the workflows from the data deposition in some space on the IFB core cluster to sending it to Galaxy, performing the analysis, getting back the results, and then we will add the submission to international databases. This is under development. The third phase will be the most difficult one, because actually we have to work with data that is sensitive. There is personal data, and the end goal is that once the data has the non-sensitive data, so the viral sequences have been submitted to the database, we can pair it with the sensitive data, and this has to be done in a certified space for hosting sensitive data or health data storage. And so this will be done at the in the CINES Center in Montpellier, And the INSERM is working with CNES in order to build this certified zone. And this is heavy work that takes already running since a few months, and that will take a few more weeks before being, or even 2 months before being ready. In the meantime, we can already make a big use of the data, because actually the sequence data is already available. No,
Jack,
in
1
conclusion,
minute.
yes, I'm at the conclusion. So basically, I showed you the progress. The phase 1 is, we can say, it's operational since several months. It has been operational very fast, and the tool, the database, has been improving progressively, and it is still improving, because now we are using it also for providing the results to the Santé Publique France, to the ministry, and so on. But also there are new actors that request the connection, because the database is very convenient. The second phase is in good way to be operational. It is we have the components, we now have to connect them, and then to organize the data flows from the platforms to IFB, and from IFB to the international centers. So this is the next challenge. And then the phase 3 is in progress. A big challenge of this project is that there is a multiplicity of the partners. We have more than 50 partners, and so this means that we have huge meetings and so on. And also that we were confronted to the trade-off between the urgency of building something that can be operational and usable, and the fact to build some solution that is robust and that fits our own criteria, IFB criteria of being open science, having all the fairness of the code, fairness of the data, and so on. And so we are navigating in this trade-off by using an agile approach. So we permanently find some temporary solution, but we already are going in the direction where we want to be within a few months. The long-term perspective, because this is a huge effort, but it's not only, well, already, would it be for COVID-19, it would be worse, but the idea also is to have general infrastructure in order to monitor any emerging infectious disease. This can also be reused for other projects in other countries, for instance, and there is a demand now to have this kind of infrastructure for African data monitoring. And then there is also a very interesting achievement here, is that everything we did here is somehow already reusable for other projects. For instance, the Abromix project, which is about antibiotic resistance. Thank you for your attention.
Thank you a lot, Jacques. So normally people are running to coffee break, but if someone has one question. Okay, so don't hesitate to ask it on the Q&A panel. And so I have one question, Jacques. So you already mentioned that, but I think that a lot of people are also involved in an environmental project that deals with viruses. And could we think that the tools that have been developed or will be developed within this project can be used also on environmental data to detect or follow the detection of viruses within this environment with this kind of samples?
Yes, definitely, yes. I didn't think about it, but all these projects, well, basically also it will be very interesting for the monitoring of viruses in animals, which is very interesting. And now INRAE is very involved in this.
Okay.
And so there is a question from Carol.
From Carol, yes.
So would you say that the workflows accelerated the development of the platform? Well, yes, Carol. Actually, first we started discussing with the sequencing platforms, and we asked them, okay, what are you using? Basically, most of them, they use the built-in workflows that were developed by the sequencing facilities with Illumina or Ion Torrent or Nanopore, and this is a black box. And what we wanted is to have to ensure the homogeneity of everything that is treated in France and everything that is treated in the other partners of Elixir. And so what Christophe Antoniewski and Gilda did is to look at what was there in Elixir, and indeed the work that had been done in particular in Germany, and also the work done in Belgium by Fred Coppens and so on, and all the workflows were already there. We have to customize them to adapt them to the specificities of the French situation, but it's a huge forward step. Yeah, so that's the great part of being a member of an international network.
Thank you, Jacques, for this answer. So So thanks to all of you for participating and for giving the talk this morning, and for all the participants to follow this talk. Thanks a lot to the organizer to help us to organize this talk, to Hervé and to Claire. And so now it's time for coffee break, and feel free to join all the different participants who all have a poster to have more information about the talk today. Bye.